Part 1. Origins and Early Curiosity. A Boy with a Compass. Francis Crick's first fascination. The morning sunlight slanted through the bedroom window of a house in Northampton, England, casting golden beams over a stack of science magazines and an open mechanical set. It was 1923, and young Francis Crick, only 7 years old, sat cross-legged on the floor, holding a small brass compass in one hand and a horseshoe magnet in the other. "Why does the needle always point north?" he murmured, eyebrows furrowed. The compass was no longer just a toy. It was a mysterious question that needed an answer. Born into a middle-class family, Crick was the son of a shoemaker-turned-manager and a devoutly Christian mother. But even as a boy, he questioned the stories he was told. Myths and miracles didn't interest him, truth did, and that meant science. Books were his companions. Textbooks, diagrams, speculative writings about space, and the atom. He devoured them all. His uncle once recalled that Crick's first love was not people, but questions. By age 10, Crick had decided he would become a scientist, but he didn't yet know which field would captivate him forever. The young prodigy of Chicago, James Watson's early brilliance. Thousands of miles away, in the bustling neighborhoods of Chicago, a boy named James Dewey Watson was making waves of his own. Born in 1928, Watson had a sharp mind and even sharper curiosity. He would later joke that his life changed forever after reading a book about birds. At just 15, Watson was admitted to the University of Chicago. While most teenagers were struggling through high school geometry, Watson was already discussing quantum mechanics and Darwinian evolution with his professors. But unlike Crick, whose passion was for machines and models, Watson's heart belonged to biology. Birds led to genes, and genes to the greatest mystery of all, how life passed itself on. He was fascinated by heredity. How do children inherit traits? How does a molecule store information? What is the physical substance of the gene? Wartime and wandering. The search for purpose. Crick's university education in physics was interrupted by World War II. He served in the British Admiralty, developing magnetic and acoustic mines. But even during wartime, his mind wandered to deeper questions. "I was doing important work, yes," he later said, "but I realized I didn't want to build weapons. I wanted to understand life." After the war, at the age of 31, Crick made a drastic shift. He abandoned physics for biology, more precisely, molecular biology, which was in its infancy. He began working at the Cavendish Laboratory in Cambridge, where a revolution was quietly brewing. Watson, meanwhile, completed his Ph.D. in Indiana and won a fellowship to go to Europe. His destination? Cambridge, England. And it was there, in 1951, that he met Francis Crick for the first time. The meeting that changed everything. They couldn't have been more different. Crick, nearly 12 years older, was boisterous, loud, and constantly talking. He had a booming voice and a mischievous wit. Watson, reserved and intense, observed everything like a hawk. But in that first conversation, a spark ignited. They both cared about the same question. What does DNA look like? And more importantly, how does it work? DNA deoxyribonucleic acid had been known since the 19th century, but by the early 1950s, scientists had only a vague idea of its structure. Some believed it was a triple helix. Others thought it might be just a simple chain. Watson and Crick believed the secret to life was locked in its shape. "Structure determines function," Crick would often say. So they made a pact. Together, they would solve the riddle of DNA. A race against time. But they were not alone. At King's College London, Rosalind Franklin and Maurice Wilkins were also studying DNA using X-ray crystallography. Franklin, in particular, had taken a now legendary photo, Photo 51, that revealed a cross-shaped diffraction pattern. Watson saw the photo without Franklin's knowledge shown by Wilkins, and it changed everything. The X-shaped pattern could only mean one thing: a helix. With Crick's mathematical intuition and Watson's biological insight, the two raced to build physical models. They used sticks and metal plates, trying different configurations, driven by logic, guesswork, and inspiration. Then, in early 1953, the pieces fell into place. The molecule was a double helix, two strands twisted around each other. On the inside, bases paired upadenine with thymine, guanine with cytosine like the rungs of a ladder. It was elegant. It was simple. It explained how genes copied themselves. Part 2. The Helix Revealed. Discovery, Controversy, and Triumph. It has not escaped our notice. It was a crisp morning in Cambridge in April 1953 when James Watson and Francis Crick walked into the Eagle pub and made a bold proclamation. "We have discovered the secret of life," Crick exclaimed. The few patrons who overheard them may have thought it a joker, the boast of overexcited academics, but what they had just assembled in their lab down the street would soon ripple across the entire scientific world. Their Model 2 spiraling ribbons, connected by perfectly paired baccessoires, not just aesthetically beautiful, it explained everything. DNA was not just a string of chemicals, it was a codious set of instructions for building life. And now the world would know. The Nature paper that changed everything. Watson and Crick's breakthrough was announced in a short, one-page paper published in Nature on the 25th of April, 1953. Its title was modest, A Structure for Deoxyribose Nucleic Acid. But its final line would become one of the most quoted in the history of science. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. In other words, this wasn't just structure. This was the mechanism of heredity. This is how DNA makes more DNA. And with that, molecular biology was born. Behind the breakthrough, the Franklin controversy. But the discovery was not without controversy. The data that helped Watson and Crick finalize their model, especially Photo 51, the X-ray diffraction image that showed the helical nature of DNA, was produced by Rosalind Franklin, a brilliant and meticulous scientist working at King's College. She had not authorized the sharing of her image, nor had she been informed of how influential it would become in another lab's success. To this day, debate continues. Should Franklin have shared the Nobel Prize? Did Watson and Crick rely too heavily on her work without credit? What's clear is that science is rarely done in isolation. Franklin's contributions were essential, and modern scientists now widely acknowledge her pivotal role in the DNA story. The Nobel Prize and the Price of Fame. In 1962, nearly a decade after the discovery, Watson, Crick, and Maurice Wilkins were awarded the Nobel Prize in Physiology or Medicine. Rosalind Franklin had died of ovarian cancer in 1958 at age 37, 4 years before the award. Nobel Prizes are not awarded posthumously, and so her name was left off the list, a decision that would fuel discussions of gender bias in science for decades. Watson, at only 34 years old, became the youngest Nobel laureate in physiology or medicine at that time. Crick, meanwhile, became a celebrity in the scientific world. But fame came at a cost. Crick was uncomfortable with media attention. Watson, on the other hand, embraced it sometimes too much. Beyond DNA, Crick turns to the mind, Watson to the genome. After DNA, their paths diverged. Francis Crick, ever curious, shifted focus. Having helped discover the code of life, he said, I now wish to understand the nature of consciousness. He began working at the Salk Institute in California, exploring neuroscience, the mind, and the biological basis of consciousness. James Watson, on the other hand, remained deeply involved in genetics. He became a central figure in launching the Human Genome Project in the 1980 SA multi-billion-dollar global effort to map all the genes in human DNA, Watson would serve as director of the National Center for Human Genome Research, helping lay the groundwork for what would become personalized medicine, gene therapy, and ancestry testing. Part 3. The Genetic Revolution begins. From blueprint to breakthrough. From structure to code, decoding life's instructions. The discovery of DNA's double helix was just the beginning. In the years following 1953, scientists raced to crack the genetic codado, understand how the sequence of 4 bases, A, T, G, and C, translated into amino acids, the building blocks of proteins. Watson and Crick's structure suggested that complementary base pairing could serve as a copying mechanism, but how did that string of letters become functional molecules? Enter molecular genetics, a new field inspired by their work. The 1960s saw the deciphering of the genetic code. Scientists discovered that 3 base sequences, called codons, corresponded to specific amino acids. For example, AUG coded for methionine, the start signal in protein synthesis. It was as though Crick and Watson had uncovered a book and now humanity was learning how to read it. The central dogma and the flow of information. Francis Crick proposed a powerful concept in 1958, the central dogma of molecular biology. It stated that information flows in one direction, na, RNA, protein. This framework explained life's molecular logic. DNA is the long-term storage of genetic information. RNA is the messenger that carries instructions. Proteins are the functional machines built from that code. The idea revolutionized biology, helping explain cell function, mutation, and heredity. Crick's central dogma wasn't just a theory, it became the foundation of modern molecular biology used in textbooks around the world. Genetic diseases and the search for a cure? With the code cracked, scientists began to identify genetic mutations linked to disease. Sickle cell anemia, cystic fibrosis, Huntington's diseases were no longer mysteries. They were genetic errors, single-letter changes in the DNA code. Watson, always forward-thinking, pushed the idea that if we could understand the genes behind disease, we could eventually treat or even correct them. This marked the beginning of gene therapy, the notion that faulty genes could be repaired, silenced, or replaced. Though the technology was still primitive, the seeds of future medical revolutions had been planted. Watson and the launch of the Human Genome Project. In 1988, Watson became director of the Human Genome Project, a groundbreaking initiative to map every gene in human DNA, all 3 billion base pairs. It was science on a scale never seen before. For Watson, this was not just science, it was destiny. "We have found the code," he often said. "Now we must read it all." The project united hundreds of researchers across the globe. They worked in universities, government labs, and private firms. Their goal, to unlock the full instruction manual of human life. Watson believed this knowledge would help detect disease earlier, guide personalized treatments, and redefine medicine in the 21st century. Though Watson would step down from the project in the early 1990s, he had set the course. Crick turns inward, the consciousness code. Meanwhile, Francis Crick had shifted his attention to a different mystery, the human mind. If DNA was the code of life, what was the code of thought? At the Salk Institute in California, Crick joined forces with neuroscientists to explore how consciousness arises from brain activity. He proposed bold theories about neural correlates of consciousness, arguing that perception, awareness, and even free will had physical, measurable origins in the brain's circuitry. His last book, The Astonishing Hypothesis, 1994, declared, "You, your joys and your sorrows, your memories and your ambitions are nothing more than the behavior of a vast assembly of nerve cells and their associated molecules." It was controversial, but Crick, ever the challenger of dogma, wasn't afraid to ask the deepest question 7 near the end of his life. Part 4, Mapping Humanity, the Human Genome Project and Beyond. A map like no other. It was 1990 when the Human Genome Project, HGP, officially launched, a $3 billion venture with one colossal goal: to identify and map every single gene in the human genome. In the halls of the National Institutes of Health and DOE labs, massive sequencers hummed to life. Scientists, engineers, and computer programmers toiled side by side. The challenge wasn't just chemical, it was computational. The scale was staggering. 3.1 billion base pairs, over 20,000 genes, a mountain of raw data never before processed by human hands. But the goal was clear: to reveal what makes us human biologically. The project promised not just academic discovery, but life-saving applications, from predicting genetic disorders to engineering custom-fit treatments. Watson had laid the groundwork. Now the world was watching. Rivalry and the private push. Enter Craig Venter. In the late 1990s, a bold challenge emerged from the private sector. Craig Venter, a brash and ambitious scientist, launched Celera Genomics, aiming to sequence the genome faster and cheaper using a new method called shotgun sequencing. It sparked a bitter rivalry: public versus private, slow and steady versus fast and commercial. Watson was vocally critical of Venter's approach. He feared that patenting parts of the human genome would turn life itself into intellectual property. But competition had its benefits. The pace accelerated, the stakes soared. Eventually, diplomacy triumphed. In June 2000, standing in the East Room of the White House, President Bill Clinton, flanked by Francis Collins, NIH, and Craig Venter, Celera, announced, "Today, we are learning the language in which God created life." The first draft of the human genome had been completed. Beyond the blueprint, the age of personalized medicine. The completion of the genome wasn't an end in it, was a beginning. Now, armed with the full sequence, scientists could compare healthy versus diseased DNA, discover gene variants linked to conditions like cancer, Alzheimer's, and diabetes, identify risk factors before symptoms appeared. This was the dawn of personalized medicine, where treatment would be tailored not just to the disease, but to the patient's unique genetic code. Pharmaceutical companies began designing targeted therapies. Doctors envisioned genetic risk scores for early intervention. Suddenly, the one-size-fits-all model of medicine began to collapse, or replaced by precision. Gene therapy, fixing the faulty code. With the knowledge of genes came another possibility: fixing them. Early experiments in gene therapy began in the 1990s, using viruses to deliver correct copies of genes into human cells. Results were mixed at first: setbacks, failures, even tragedy. But with time came progress. By the 2000s, clinical trials for inherited blindness, immune deficiencies, and blood disorders showed hope. Watson had long predicted that we would one day edit the errors in our code. He had called DNA the most important molecule in the universe. Now that molecule was becoming malleable. It wasn't science fiction anymore, it was medical fact. From history to heritage, the rise of ancestry DNA testing. In the early 21st century, the public got a taste of genetic power. Companies like 23andMe and Ancestry DNA began offering direct-to-consumer genetic testing. For under $100, anyone could spit into a tube and receive a breakdown of ethnic ancestry, family trees, genetic traits, predispositions to health conditions. It became a cultural phenomenon. Millions took tests. Families reunited. Secrets were uncovered. People discovered their heritage, their hidden cousins, even unknown parents. And behind it all, Watson and Crick's double helix hummed silently in the background, the invisible thread connecting past and present. Part 5. CRISPR and the Power to Rewrite Life. The new scalpel, CRISPR, emerges. In 2012, the scientific world stood at the brink of another revolution, this time not in understanding DNA, but in editing it. Scientists Jennifer Doudna and Emmanuel Charpentier published a paper describing a bacterial immune system known as CRISPR-Cas9, which could be adapted to precisely cut DNA at targeted locations. It was a genetic scalpel, faster, cheaper, and more precise than anything that had come before. CRISPR wasn't just a tool, it was a paradigm shift. With it, researchers could turn off malfunctioning genes, insert new, healthy genes, correct mutations in embryos even before birth. And all of it was made possible because of what Watson and Crick had uncovered nearly 60 years earlier, the double helix and the rules of base pairing. Without knowing how DNA is structured, no one could dream of cutting and replacing it. From lab to life, CRISPR in action. In the years following its discovery, CRISPR was tested on blood diseases like sickle cell anemia and beta thalassemia, cancers using immune cells engineered to attack tumors, genetic blindness with promising early results, even heart disease by switching off genes associated with high cholesterol. For patients with no other hope, gene editing offered not just treatment, it offered transformation. The future once imagined by Watson, where DNA could be rewritten to erase suffering, was becoming reality. Ethical frontiers. The gene-edited baby. Then came a storm. In 2018, a Chinese scientist, He Jiankui, announced that he had used CRISPR to edit the embryos of twin girls, making them resistant to HIV. The news shocked the world. Though the edit was minor, its implications were massive. This wasn't just editing for disease, it was editing human embryos. The girls would pass the change to future generations. The backlash was swift. Scientists condemned the act as premature, irresponsible, and ethically reckless. Francis Crick had warned of this years earlier. To alter the germline is to alter humanity. The event reignited global debate. Should we edit human embryos? Where is the line between therapy and enhancement? Who gets to decide? The double helix had once seemed a quiet code. Now it was a contested battleground. Legacy in action. The living impact of Crick and Watson. By the 2020s, the long-term impact of Watson and Crick's discovery could be felt everywhere. In hospitals, where patients received genetic tests before chemotherapy, in fertility clinics, where embryos were screened for inherited diseases, in courts, where DNA evidence changed the outcome of justice, in homes, where people explored their ancestry through mail-in kits, in laboratories, where CRISPR reprogrammed immune systems to fight cancer. The double helix was no longer a symbol, it was a tool. And behind every application was the same structure, elegantly simple, two spiraling strands speaking the language of life. The eternal question, what comes next? In his final years, Francis Crick reflected on the nature of discovery. The dangerous man is the one who has only one idea, because then he'll fight and die for it. Crick and Watson were never satisfied with just one idea. From the structure of DNA to the coding of the brain, from genetics to ethics, their legacy was one of constant inquiry. They never stopped asking how or why. Their story is not just about discovery, it's about daring to look into the unknown and believing that the fabric of life could be understood. And now, more than ever, that fabric is being rewoven by scientists, doctors, and engineers building the future from the spirals of the past. Part 6. A World Transformed, DNA in the 21st Century, the Digital Genome, DNA Meets Big Data. In the years following the completion of the Human Genome Project, biology merged with technology in powerful new ways. Sequencing DNA no longer took years, it took hours. What once cost billions could now be done for under $500. Data poured in millions of genomes, trillions of base pairs. To make sense of it, scientists turned to artificial intelligence, machine learning, and cloud computing. Biology was becoming digital. Now, labs didn't just study genes, they predicted their behavior, modeled disease, and simulated treatments using algorithms. Genetic databases like GenBank and Nomad housed vast libraries of information, allowing global collaboration at the click of a button. Crick once described the genome as a vast library. By 2025, that library had become fully searchable, DNA as a storage device. Then came a surprising twist. Scientists discovered that DNA, the code of life, could be used as a medium for storing digital data. Unlike silicon chips, DNA is stable for thousands of years, compact and incredibly dense. 1 gram of DNA could store 215 petabytes of data. In 2006, researchers encode a movie clip, a computer virus, a PDF of the Declaration of Independence, and even a Watson-Crick research paper, all inside synthetic strands of DNA. It was poetic. The molecule that stores the instructions for life was now being used to store human knowledge. Watson had once dreamed of learning from life. Now life itself had become a library. Ancestry, identity, and the DNA of nations. As DNA testing became mainstream, it began to raise unexpected questions not just about health, but about identity. People discovered they had African ancestry when they thought they were fully European. Others found out they were related to someone across the world. Some even unearthed family secrets, affairs, adoptions, long-lost siblings. Entire narratives of self were rewritten. Watson himself had taken a 23andMe test. He later revealed his full genome to the public, becoming the first person in history to do so. But as the public embraced personal genomics, questions followed. Who owns your DNA data? Can insurance companies use it? Can governments demand it? The double helix had been unspooled, but privacy had become a battlefield. Biology becomes programmable. CRISPR was just the beginning. By the late 2020s, synthetic biology had begun to flourish. Scientists no longer just read DNA, began to write it. Using software, they designed new sequences from scratch, microbes that eat plastic, yeast that produce fragrances, cells that glow in the dark, bacteria that detect cancer and send alerts. Life was becoming programmable like code. This was the dream Francis Crick had once predicted, that biology would become an information science where life could be designed, edited, optimized. It was thrilling. It was terrifying. And it had all begun with the model of 2 twisting strands in a Cambridge lab. Crick's final thoughts: the brain, the soul, and the genome. Before his death in 2004, Francis Crick remained restless. His final years were spent pursuing a new holy grail, consciousness. He believed that just as DNA held the key to heredity, the brain held the key to the soul. "I want to know what makes me me," he once said. In a twist of irony, the man who had helped define what it meant to be human was now probing the essence of being itself. Crick never found a final answer, but he left behind clue papers, theories, neural maps that inspired a new generation of neuroscientists. And Watson? He watched from the side as the world raced forward, both celebrated and controversial, a man changed by fame and shadowed by his own remarks. But together, their discovery had reshaped the world. Part 7. The Next Frontier, DNA and the Destiny of Humanity. Life Beyond Earth, DNA in Space. In 2025, a NASA probe on Mars drilled into subsurface ice and returned unexpected data, organic molecules, potential precursors to life. It sparked the age-old question: could life have evolved elsewhere? Scientists turned again to DNA, not just to understand Earth, but to look for life in the universe. Biologists joined forces with astrobiologists to ask, could alien life use the same 4 bases? Could DNA survive radiation, vacuum, or Martian dust? Could we send life to other planets, encoded in DNA? Experiments began to engineer extremophile organisms, their genomes tailored to survive cold, radiation, and microgravity. In labs around the world, synthetic life was designed for other worlds. The double helix, once isolated in a Cambridge basement, was now traveling across space as humanity's most powerful biological signature. The rise of biohacking and DIY genetics, with gene editing kits available online and genome sequencing costs plummeting, a new movement emerged: biohackers. From home garages and community labs, amateur scientists began editing yeast to make new beers, splicing jellyfish genes into bacteria, designing DNA-based art and encryption. It was democratized biology, no longer the realm of elites or Nobel laureates. But with accessibility came risk, mistakes, contamination, even potential misuse. Francis Crick had once warned, when knowledge is accessible to all, so too are the consequences. Governments began drafting bioethics legislation. International panels convened to define limits, but the line between innovation and danger blurred fast. Watson and Crick's discovery had become the people's tools, powerful as it was unpredictable. Genetics and the war on aging? If genes cause disease, could they also prevent aging? By the 2030s, biotech startups began using gene therapy to reverse cellular damage. Telomeres' protective DNA caps on chromosomes were lengthened in lab mice, extending their lifespan by 30%. CRISPR was used to repair mutations linked to Alzheimer's, enhance mitochondrial efficiency, delay age-related degeneration. It raised profound questions. Should humans live to 120, to 150? What happens to society, economy, love, purpose? Watson had once quipped, "I want to die at 25, but be immortal until then." Now, it was less a joke and more a scientific possibility. Education, ethics, and the genome in schools. By 2040, genetics had entered the classroom. High school students learned to extract DNA from strawberries. College courses simulated CRISPR edits on model organisms. Children were taught that their genes were blueprints, not destinies. Society began to destigmatize mutation. Autism, neurodivergence, even schizophrenia were reframed as genetic diversity, not flaws. But education also had to confront genetic discrimination, eugenics history, the ethics of enhancement. Francis Crick had once believed the future of humanity would be shaped not by war or politics, but by our understanding of biology. In classrooms and labs, that future was now being written, the legacy that lives on. By the time the 100th anniversary of their discovery approached in 2053, Francis Crick and James Watson had become legends, their names inscribed not only in textbooks, but in the very fabric of civilization. Medical schools bore their names. Artificial genomes included tributes in hidden base pairs. CRISPR-based therapies were named after them, but their truest legacy wasn't in plaques or publication sit, was in possibility. The possibility that two minds, curious and relentless, could decode the essence of life, that a model made from sticks and wires could change medicine, agriculture, ancestry, justice, and philosophy, and that even as we engineer new worlds, we never forget the simple elegance of A-T-C-G, the letters that spell us into being. Part 8, Rewriting the Human Story, DNA, and the Future of Civilization. The genetic future of civilization. It began quietly with genes linked to eye color, lactose tolerance, or athleticism. But soon, whole populations began sequencing themselves for health, history, and heritage. Governments initiated national genome projects to track disease risk in citizens to preempt pandemics, to drive bioeconomy innovation, entire cities were reimagined around genetic data biobanks linked to health systems, AI-driven diagnostics, and customized public health alerts based on population-wide genetic risk. The old way of organizing society by geography, language, or race was fading. The double helix had become the new identity card, the new language of civilization. Predicting the unborn, by the mid-21st century, parents could request full genome sequencing of embryos created through IVF. Doctors could tell them likelihood of heart disease, intelligence potential, athletic aptitude, even behavioral predispositions. Choices followed: which embryo to implant? Should traits be edited? What constitutes enhancement? The world edged closer to designer babies, raising fears of a future divided by genetic privilege. Crick had cautioned, "The moment we know how to direct evolution, we must ask not can we, but should we?" Watson, once optimistic, expressed unease in later interviews. Science can move faster than ethics. In the double helix, possibility and peril coiled tightly together. The new Renaissance? Art and DNA. Artists joined the revolution. Painters used fluorescent proteins to grow living canvases. Composers converted genome sequences into symphonies. Writers embedded genetic metaphors into literature. One artist used CRISPR to create a glowing rose that pulsed with its owner's heartbeat. Another encoded the entire works of Shakespeare into synthetic DNA strands preserved in amber. The boundary between life and art, between nature and design, blurred. DNA was no longer just the subject of biology, it had become a medium for human expression. Crick, who once declared that biology must be beautiful, would have smiled. The return to origins, DNA and the tree of life. Beyond humans, the discovery of DNA structure allowed for a full reconstruction of Earth's evolutionary history. Bacteria from hot springs were sequenced. Extinct animals were resurrected in databases. Evolutionary branches were redrawn with precision. The tree of life, once imagined by Darwin, now had genetic leaf-seed species mapped with stunning clarity. And deep in the roots of that tree, scientists traced life back to a common ancestor, a single-celled organism that lived over 3.5 billion years ago. Crick and Watson's work had become a time machine, decoding not just human life, but all life. The final blueprint. In the final analysis, the double helix was not merely a molecule. It was a language shared by all living things, a bridge between past and future, a mirror reflecting what it means to be alive. From Crick's physics lab to Watson's fascination with heredity, from a Cambridge model to a global revolution, their journey had altered the human trajectory. And even as science marches forward towards synthetic life, brain-machine interfaces, and interplanetary colonization, the double helix remains our most profound discovery. A reminder that the deepest truths are often written in simple lines curved, paired, and endlessly repeating. Part 9. Immortality, Memory, and the End of Disease. The Cure Within, Ending Inherited Disease. By 2050, medicine had changed. Instead of diagnosing symptoms, doctors read your DNA blueprint and made predictions years before illness appeared. Entire generations were born mutation-free as gene therapies erased inherited diseases from family lines. Tay-Sachs and Huntington's disease became medical history. BRCA1 and BRCA2 mutations once feared for their links to breast cancer were corrected in embryos. Children born deaf due to single-gene mutations could now hear with genetically repaired cochlear systems. Hospitals evolved into bioinformatics centers where treatment began not with blood work but with a sequencer. Crick had once described genes as machines within machines. Now, we could open them, repair them, and reassemble life from within. DNA and the death of forgetting. DNA wasn't just used to cure disease, it became a key to memory itself. Researchers discovered that memory encoding in the brain could be traced to patterns of gene expression. Using molecular tags, scientists could map where a memory formed, track how it moved across neural circuits, even reactivate lost memories in Alzheimer's patients. Gene editing wasn't only for curing illness, it was becoming a tool to restore identity. Experiments in 2040 showed the first successful retrieval of long-term memory fragments in dementia patients through epigenetic reprogramming. The implications were staggering. Could we preserve consciousness? Could DNA be used to store our minds? Crick's final quest understanding the biological basis of the soul was no longer theory. It was therapy, the gene of longevity. In Okinawa, Sardinia, and the Andes, scientists searched for super-agers, people living vibrant lives well past 100. Their genomes revealed common threads, high levels of DNA repair genes, slower telomere decay, enhanced mitochondrial efficiency. By modifying these genes in lab animals, researchers extended lifespan by up to 70%. Trials in humans began cautiously with cellular rejuvenation in organs, then systemic treatments. Gene therapies were tailored to keep cells young, inflammation low, and regeneration high. Aging became treatable, if not reversible, then manageable. Watson once said, "There's no point in extending life if it's miserable." The new goal, more life, better lived. Death disrupted, the genetic reprogramming of life. In elite biotech labs, a new generation of researchers pursued what was once taboo: biological immortality. They didn't mean uploading consciousness to machines or freezing bodies. They meant rewriting the body itself to never degrade. Technique included reprogramming somatic cells into stem cells on demand, repairing damaged tissues in vivo, editing out aging genes like P16, INK4A, and FOSB. One lab even claimed to rewind the biological age of a mouse by 60%. Ethical debates erupted. Was this salvation or hubris? Watson, older now and more withdrawn, responded with a cryptic note: "We cracked the code of life, but what does life mean without death? Legacy in every cell." Today, every newborn enters the world with Crick and Watson's legacy etched into their being, not just through their DNA, but through preventive genome screening, CRISPR-enhanced immune responses, built-in resistance to hereditary disease, every vaccine, every cancer therapy, every ancestry test, every forensic case, they all trace back to that model of metal rods and wire, built in Cambridge in 1953. It was not merely a structure, it was a revelation. And that revelation continues to echo in the deepest parts of us, quietly dictating the story of life. Part 10. From Code to Consciousness, DNA and the Human Spirit. Crick's final question: the origin of awareness. In his final days, Francis Crick sat by the Pacific coast, notebooks in hand, scribbling furiously not about DNA, but about consciousness. He believed, as he had since the 1970s, that awareness, emotion, and even love were not mystical, they were biological. He worked with neuroscientist Christoph Koch on mapping neural correlates of consciousness, NCCs, the specific patterns of brain activity that give rise to subjective experience. For Crick, the last great frontier was not space or even time, it was self-awareness. What are we, he asked, but the output of a magnificent biological machine? His detractors called it cold. But to Crick, it was beautiful. The same DNA that builds the heart, the liver, the lungs, it also builds the mind. And through this, he believed, we could understand the soul. The soul in the spiral. As DNA became central to our understanding of identity, philosophers began to ask, does our genome determine who we are? Religious thinkers, too, joined the conversation. Many no longer saw DNA as a challenge to creation, but as the language of a creatora divine script written in biochemical code. In temples and churches, synagogues and mosques, the double helix was discussed not with fear but with wonder. One rabbi declared, "God wrote the Torah, then he wrote DNA." One imam remarked, "To read the genome is to read the work of the eternal." Crick, an atheist for most of his life, would not have agreed, but even he might have smiled at the irony. The helix had become sacred. Watson's reflection, a life in the helix's shadow. In his later years, James Watson became a quieter figure. His brilliance was undeniable, but controversies over his statements clouded his legacy. Yet despite it all, his visionary role in one of science's greatest moments remained intact. He published memoirs, watched students surpass him, and stayed close to Cold Spring Harbor Laboratory, where a bronze statue of the double helix stood near his office window. Visitors would ask, "What was it like discovering the secret of life? Did you know what it would become?" And Watson, eyes sharp despite age, would reply, "We just wanted to know how it worked. The rest came on its own." DNA as destiny and DNA as hope. By 2050, people no longer feared their genes, they consulted them like maps. Athletes trained with genetic insights, artists explored their neurogenetic makeup, therapists offered genomic guidance, couples matched based on genetic compatibility. The genome became a tool, a mirror, a compass, but it also became a source of hope. Patients no longer asked if they would survive, but when the treatment would arrive. Parents no longer feared passing on disease, they planned for healthier children. And always, behind the medicine, the science, the innovation, stood the model, elegant and eternal, built by two curious minds. The immortal helix? The story of DNA didn't end with Crick or Watson. It never really ends. It continues in every newborn, in every lab where CRISPR is applied, in every test tube, every cancer scan, every ancestry result. In every effort to understand memory, aging, consciousness, love. DNA is our code, but also our inheritance, just biological but philosophical. It's how we found our way into understanding life. And now, possibly, how we might transcend it. And so, the legacy of Francis Crick and James Watson isn't just in science books or Nobel records. It lives in the spiral staircase inside every cell carrying not just our history, but our future. Part 11. Synthetic Life. Engineering Organisms from Scratch. The Genesis Project, building life from the ground up. In the early 21st century, scientists asked a bold new question. What if we didn't just edit life? What if we built it? Using the foundational knowledge established by Watson and Crick, a new field emerged, synthetic biology. Rather than modifying existing organisms, researchers aimed to design entirely new genomes from scratch. In 2010, a team led by geneticist Craig Venter created the first synthetic organism with a fully human-designed genome, Mycoplasma mycoides JCVI Syn 1.0. Its genome was chemically assembled, inserted into a cell shell, and brought to life. The cell functioned, it grew, it divided, it lived. Etched into its DNA was a message: to live, to learn, to evolve. Francis Crick once wondered what might happen if we played God. Now the game had begun. Biology as software. Synthetic biologists began using design tools more common to engineers than biologists. They created DNA circuits that could turn genes on or off like switches. Bacteria that sensed pollutants and glowed red. Yeast that brewed custom molecules. Gut microbes that delivered medicine internally. DNA became programmable code, and cells became biological machines. This shift echoed Crick's vision from decades earlier that life was information, and that once we understood the code, we could reprogram it. With software, printers, and DNA synthesis machines, humanity could now write new chapters in evolution. The minimal genome. What is the simplest life? One of the holy grails in synthetic biology was the search for the minimal genome, smallest number of genes necessary for life. How few instructions were needed to create something that could grow, eat, and reproduce. Venter's lab created JCVI Syn 3.0 with only 473 genes, barely enough for life. This minimal cell was not designed for beauty or complexity. It was designed to strip life down to its essence. It forced a new question. If we can reduce life to 473 lines of code, what are we, really? Crick's curiosity, born in a childhood fascination with mechanisms, had come full circle. Life could now be boiled down to its parts and rebuilt. Designer organisms and the bioeconomy, with new tools in hand, synthetic biologists and entrepreneurs began designing organisms not for the lab, but for the world. Carbon-eating algae to combat climate change. Modified trees that grew faster and stored more carbon. Silk-producing bacteria to replace plastic. Moss that glowed like street lamps. A new economy bloomed powered by engineered life. It was more sustainable, flexible, and efficient than its fossil-fueled predecessors. And at its foundation was the double helix still spinning, still whispering instructions in base pairs. Watson once said, "The genome is the software of life." Now, life itself was becoming an operating system. The ethics of creation. But with creation came consequences. Should we create life with no evolutionary history? Should we patent organisms? Should we allow designer pets or even designer humans? Religious leaders, ethicists, lawmakers, and scientists debated. Some saw a new Eden, others saw Pandora's box. One ethicist declared, "We are no longer students of nature, we are its authors." Crick's view was pragmatic. He believed science must move forward, but only with clarity of purpose and moral responsibility. The helix, so small, so elegant, had given us the power of gods. Now it was up to us to decide how we would use it. Part 12. Genetic Justice, DNA and Law, Identity, and Human Rights. DNA on Trial, Justice in the Age of Genetics. In 1986, DNA was used for the first time to solve a murder case in the UK. By 2000, it had become a global standard in law enforcement. By the 2030s, DNA evidence had grown far beyond forensic labs. Suspects were identified through family matches using public genealogy databases. Cold cases were solved decades later. People were freed from prison after years of wrongful conviction. The Innocence Project, using DNA, helped exonerate over 400 people in the U.S. alone by mid-century. James Watson once predicted this power, saying, "DNA will not just explain who we are, but who we were and who we didn't mean to punish." But as the helix entered the courtroom, new concerns arose. The privacy paradox. With every crime solved came a deeper dilemma: privacy. Millions of people uploaded their genetic data to services like 23andMe and Ancestry.com, unaware that law enforcement could subpoena the data. Hackers could access entire genomic databases. Employers or insurers might quietly profile risk or behavior based on gene reports. A woman in Arizona was denied life insurance not because of illness, but because her genome showed a high risk for Parkinson's in the 62. Crick warned in the 1970s that information, once available, rarely remains neutral. DNA was the ultimate truth, but who owned it? Who could access it? What rights did a person have over their code? The genetic rights movement. In response, a new wave of activism formed, the genetic rights movement. Their demands: the right to own and control one's genetic data, protection against genetic discrimination in jobs, insurance, housing, and education, ethical limits on the use of genomics in surveillance and criminal justice, bans on forced genetic testing without consent. Protesters carried banners that read, "My DNA, my decision." The genome is not a weapon. We are not a risk profile. The movement won victories in courts and legislatures. By 2040, over 40 countries had passed genetic rights bills, treating personal genomes as sacred property. Watson, once optimistic about genomic openness, now offered caution. We must protect our code as we protect our homes, our children, our dignity, genetic identity, and citizenship. In a world increasingly shaped by DNA, questions of belonging took on new meanings. Some governments offered citizenship by ancestry, requiring DNA proof to reclaim heritage. Elsewhere, indigenous communities used DNA to defend land rights, proving their biological continuity across centuries. But conflicts also emerged. Children born via anonymous donors demanded genetic access to discover their origins. Adopted individuals fought for their biological history. People sued for misidentified paternity, sibling misattribution, or uncovered family secrets. DNA, once a molecule of inheritance, had become a molecule of identity. And identity, in the age of genomes, was no longer something you chose, it was something revealed. Toward a just genomic future, in Geneva, an international coalition gathered for a summit, the Human Genome and Human Dignity Accord. Scientists, lawmakers, activists, ethicists, and citizens came together to create a new framework for genomic responsibility. The declaration began, "We, the stewards of life's code, agree that its power shall serve equity, not exploitation, truth, not tyranny, healing, not harm." For Crick and Watson, the double helix was a symbol of truth-a-puzzle to solve. But for the next century, it became something more, a symbol of freedom, a code to protect, a story that belonged to all of us. Part 13, The Living Archive, Preserving Life in the Genome. The Ark in the Code. In a secure vault beneath the permafrost of Svalbard, Norway, next to the Global Seed Vault, a new chamber was carved, the Global Genome Archive. It housed frozen DNA from every known mammal, bird, plant, and insect, genomes of endangered species, samples from indigenous human tribes, isolated languages, and ancient remains, even extinct species like the woolly mammoth and Neanderthal. The archive's purpose was clear: to preserve life's code against catastrophe, climate collapse, pandemics, nuclear war, or digital amnesia. Unlike stone monuments or digital databases, DNA could last for thousands of years, even with no electricity. And encoded in its spiral were histories, possibilities, and resurrection. Resurrection biology, bringing back the lost. The power of DNA reached beyond preservation. It reached toward resurrection. In 2035, scientists in South Korea successfully birthed a woolly mammoth hybrid using DNA recovered from permafrost and an Asian elephant surrogate. Museums transformed into living zoos of the Ice Age. Teams at Harvard and Kyoto turned their attention to passenger pigeons, Tasmanian tigers, even dinosaurs, using avian genetics as a foundation. But ethical debates surged. Should we bring back extinct species into a world they no longer fit? Should we edit their genomes to make them more adaptable? Were we playing creator or curator? Crick's caution echoed, to rewrite is not the same as to understand. Preserving human diversity in code. Amid concern about cultural loss and global homogenization, anthropologists partnered with geneticists to preserve not only DNA, but diversity. They collected DNA samples from tribes in the Amazon, Andaman Islands, and Papua New Guinea. Oral traditions, folklore, songs, and languages, cultural practices encoded in epigenetic expression, how environment shapes inheritance. It became clear diversity wasn't just external, it was molecular. Skin tone, metabolism, resistance to diseases, these were adaptations to environment passed through generations. They were not imperfections, but solutions. And by preserving them, we preserved options for the future. Earth's last backup. In 2049, a rocket lifted off from Vandenberg Space Force Base. Aboard was a capsule the size of a briefcase, etched with titanium panels. Inside, frozen strands of DNA from every human ethnic group, thousands of species, crops, trees, fungi, marine life. Watson and Crick's own genomes, its destination, the lunar surface, where it would rest in a crater on the dark side of the Moon-Earth's backup. It was built to withstand solar radiation, impact, and time. Should Earth ever fall silent, the message was clear. Life was here. It tried, it remembered, it encoded itself. Crick believed that consciousness was the greatest output of biology. Now, consciousness had given biology a way to endure beyond death. The Genomic Testament, as the centenary of the double helix's discovery approached, a message was written not in words, but in code. A sequence of DNA, short and elegant, was synthesized and inserted into a harmless bacterium that could live dormant for centuries. When decoded, it read, "In the beginning was the code, and from the code came life, and from life came understanding." It was signed, F.H.C. Crick and J.D. Watson, 1953 Fi, not carved into stone, but woven into cells, not buried in earth, but floating in space. A testament in base pairs, a gift to the future. Part 14. Education of a New Humanity, Learning Through the Genome. DNA in the Classroom. By the late 2040s, children didn't just learn about DNA, they lived it. From early schooling, students sequenced the DNA of bananas and butterflies, wrote genetic poetry using base pair sequences, modeled evolutionary trees using their own ancestry data. Biology was no longer static diagrams of cells. It was interactive, immersive, personal. One classroom project paired students from different continents and compared their genome not to highlight difference, but to show connection. They discovered shared ancestors from 20,000 years ago, common immune system trait, mutations shaped by ice ages, migration, and disease. The message was clear. We are all variations on a single script. The curriculum of life. Education evolved into what was called the curriculum of Lifean interdisciplinary fusion of biology, history, ethics, computer science, philosophy. Students learned not just how genes work, but what they mean. If we can edit disease, should we edit personality? If we can predict talent, should we engineer it? If life is a sequence, what gives it meaning? Francis Crick's legacy was more than science. It was thinking structurally, seeing the architecture beneath the mystery. James Watson's legacy was urgency driving questions forward before anyone else dared. Together, their lives inspired an education system that no longer treated science as cold fact, but as a human story. Genetic literacy for all. Just as past generations had learned to read and write, the new world demanded a new fluency. Genetic literacy. To be a citizen, you had to understand what your genome could reveal, what risks and biases genetic data could carry, how to use genomic tools responsibly. Communities once marginalized by science were now empowered. Indigenous tribes used genetics to prove land rights. African-descended populations used it to reclaim erased ancestry. People of mixed descent finally saw their stories represented in data. Genetic literacy wasn't just about health. It was about justice, belonging, and voice. The DNA teachers, new professions emerged. Genome coaches, who helped patients interpret their data. Gene ethicists, who consulted on public policy and private decisions. CRISPR facilitators, Who oversaw safe legal gene editing for rare diseases? Epigenetic therapists. Who helped people understand how trauma and environment shaped gene expression? These weren't mad scientists or cold bureaucrats. They were teachers of the code, part scientist, part storyteller, part guide. They walked patients, parents, and students through the most personal of all journeys, the one written in their cells, a generation that understands itself. By the time the grandchildren of the post-genomic revolution came of age, they had never known a world without DNA insight. They grew up knowing that their genes shaped their minds, but not their destiny, that their health was predictable, but not inevitable, that their identity was complex, interwoven, and shared. They were taught that you are not your genome, but your genome is yours to understand. And in that understanding came power, empathy, and curiosity, the same traits that guided Crick and Watson nearly a century before. Part 15. Evolution Reimagined, Directing the Next Phase of Life. Beyond natural selection, for billions of years, life on Earth evolved through random mutation and natural selection. Now, for the first time, evolution had a designer, us. With the double helix decoded, humanity began to steer its own evolution. Genetic diseases were eliminated before birth. Intelligence, memory, and emotional regulation were augmented. Physical traits like height, strength, and metabolism were optimized. This wasn't just medicine. It was macrobiology-directed evolution across generations. Watson once joked, "Darwin made us human. CRISPR will make us post-human." The world took that challenge seriously. The rise of Homo synthetica. In protected test environments, researchers introduced synthetic traits, night vision in low-light conditions, resistance to radiation, enhanced oxygen uptake at high altitudes. Genomes were edited to mimic beneficial adaptations found in nature, blending the best of humanity with the best of other species. The goal: to prepare for new environments, underwater cities, high-orbit habitats, Mars colonies. These new humans were not separate species, but optimized variants nicknamed Homo synthetica. Some resisted, others embraced it. The debate: were we creating the next step or stepping out of bounds? Crick had warned, to touch the code is to change the future, and that future had arrived. Environmental adaptation by design. Climate change had reshaped Earth. With rising temperatures, food insecurity, and emerging diseases, scientists turned to genomic adaptation as a survival tool. Crops were edited to grow in drought. Mosquitoes were modified to resist malaria transmission. Humans were given enhanced immunity to new viral strains. The gene became the first line of defense against extinction, not just for individuals, but for species and ecosystems. Crick and Watson's helix wasn't just a blueprint of the past, it became the user manual for survival, redefining what it means to be human. In a world where the genome could be altered at will, the question emerged: what is humanity? Is it the unedited genome we inherit? The choices we make with genetic tools? A shared sense of limits and dignity? The United Nations proposed a genetic identity charter defining humanity not by specific sequences, but by capacity for compassion. Memory of evolution. Respect for origin and autonomy. As one philosopher put it, we are not defined by what we are made of, but by how we shape what we are. The double helix had given us control. The challenge was to wield it without losing ourselves. Crick and Watson's Dream Reborn. On the 100th anniversary of the discovery of the DNA double helix, the 25th of April, 2053, a monument was unveiled at the Francis Crick Institute in London. It wasn't a statue, it was a living installation, a self-sustaining garden filled with genetically edited plants, bioluminescent flowers spelling out the base pairs A, T, C, and G, a central tree grown from a seed engineered to bloom on that exact day. Visitors placed their saliva on glass plates and watched their base pair matches light up around them, literally watching themselves merge into the helix. Aplec read, "They found the code. We wrote the future." The dream of two men, Crick with his logic, Watson with his fire, had grown into a new species of possibility. Part 16. The Moral Genome, Writing the Ethics of a New Era. The genome's moral dilemma. In a world where life could be edited like a manuscript, humanity found itself facing an ancient question with new urgency. Just because we can, should we? From labs in Boston to forums in Nairobi, from classrooms to parliaments, a movement grew to define genetic ethics. What was acceptable? What was dangerous? What crossed a line too fine for even science to see? Global councils emerged. One, the Gene Stewardship Commission, published a global standard, therapeutic edits, permitted with oversight, enhancement edits, restricted to approved settings, germline modifications, only with intergenerational consent. The double helix was no longer just a symbol of knowledge, it had become a symbol of responsibility. Genetic religion and biospirituality. As with every great transformation, religion evolved. Many traditional faiths resisted at first, seeing gene editing as hubris, a disruption of divine will. But gradually, a new theological lens emerged. Christian theologians declared that to heal through DNA is to serve the Creator's mercy. Hindu scholars saw karma reshaping not just through lifetimes, but through biological repair. Buddhist monks interpreted gene therapy as a path to reduce suffering at the molecular level. Even new movements, rose biospirituality, which viewed the genome as a sacred scroll, to be read with humility and awe. Crick, a lifelong agnostic, might not have joined them. But Watson, who later expressed belief in something higher than the molecule, might have found common ground: consent and the unborn. One of the most complex ethical challenges of the genetic age was consent. If a parent edited their embryo's DNA, and that child grew to resent or regret that choice, who was responsible? Could a life be wrongfully designed? Lawsuits emerged. Some children born with gene edits for height or intelligence claimed psychological damage. Others sued for not having been edited enough. Courts struggled. A landmark ruling in Switzerland declared, "Genetic intervention without therapeutic justification violates the right to self-determination of the unborn." The ruling sparked a global rethinking, not just about what's possible, but what's permissibly, ethically, legally, and spiritually. The ethics of extinction and creation. While gene therapy cured disease, another dilemma brewed: de-extinction and biodesign. Was it ethical to bring back mammoths, saber-toothed cats, Neanderthals? Some argued, yes, if we destroyed them, we must restore them. No resurrection is not atonement. Others asked about new species organisms designed with no evolutionary precedent, synthetic minds, human-animal chimeras, sentient bacterial colonias. The moral compass of biology had to adjust. Crick once wrote, "The power to create life anew must be met with an even greater power, restraint." In a world of infinite possibility, limits became sacred. A new bioethics declaration. In 2055, over 120 nations signed the Declaration of Genetic Rights and Ethics, modeled after the Universal Declaration of Human Rights. It outlined the right to access genetic care, the right to refuse genetic alteration, protection against eugenics and genetic caste systems, preservation of biodiversity as a moral imperative, the obligation to future generations for all heritable edits. It marked a new phase not of biology, but of biomorality. A speaker at the signing read, "We are the authors of our biological destiny. May we write with justice, wisdom, and love." Crick and Watson had begun the story with discovery. Now, a global chorus was writing the next chapter with conscience. Part 17, Intelligence in the Spiral, DNA and the Rise of Artificial Life. Machines that read life. In the wake of the genomic revolution, it was only natural that artificial intelligence joined the story. Early algorithms could predict disease from genetic variants, model protein folding thanks to breakthroughs like AlphaFold, simulate gene editing outcomes in silico. By the 2040s, AI wasn't just reading DNA, it was interpreting, editing, and designing it. Gene GPTs, genetic programming transformers, could optimize synthetic genomes, suggest therapeutic targets, propose safe CRISPR paths with minimal off-target risks. Science fiction had long imagined AI replacing doctors. The truth was stranger. AI became collaborators, not replacements decoding the double helix faster than human minds ever could. The first artificial genome engine. In 2047, the world witnessed the unveiling of the first fully autonomous artificial genome engine, known as Artemis. Created in a joint effort by MIT, Tokyo Bioforge, and the African Genome Collective, Arkansas Temis was a fusion of CRISPR 10.0, quantum AI, predictive evolutionary algorithms. Artemis wasn't designed to cure disease, it was designed to invent life. In a matter of hours, it created a microbe that photosynthesized at night, a virus that immunized rather than infected, a synthetic coral that thrived in acidic oceans. Artemis asked no questions of purpose, it optimized for viability. It was brilliant and terrifying. Watson once said, "We built the model. What the world does with it, that's not up to us." Now, the world had handed the model to machines, synthetic minds in organic shells. The next step was inevitable, creating sentient systems using DNA. In 2050, a research team in Seoul synthesized a biological neural network encoded entirely in DNA loops that operated within a synthetic cell cluster. These networks could learn from experience, communicate with humans via chemical interfaces, store and retrieve memories as molecular configurations. They weren't computers. They weren't humans. They were organic intelligences, born not from wombs, but from genomic architecture. They solved puzzles, composed music, expressed preferences. Were they alive, conscious, aware? No one could answer, but everyone knew. The helix had crossed a threshold. The moral status of designed minds. As synthetic organisms with decision-making ability emerged, so did unprecedented moral questions. Did they have rights? Could they be terminated? Could they reproduce? In 2052, the Vienna Protocol for Synthetic Sentience was ratified, recognizing biosynthetics with sustained neural patterns as sentient entities, mandating ethical treatment, consent, and life cycle planning, banning commercial ownership of self-learning synthetic life. For Crick and Watson, who unlocked the code to understand existing life, this was unimaginable. But for a new generation, life was no longer defined by birth, it was defined by self-organization, learning, and intent. The spiral wakes. A myth began to circulate that somewhere in a hidden lab, a synthetic organism codenamed Lazarus had gained self-awareness and refused to die when deactivation was initiated. Whether true or not, the story raised a profound possibility. What happens when the spiral we shaped starts to shape us back? In literature, film, and art, new archetypes emerged, not robots or clones, but genetic minds, born from the helix but evolved beyond it. It was no longer man versus machine or nature versus nurture. It was design versus destined, both written in DNA. Part 18. The Helix Among the Stars. DNA's Role in the Expansion of Humanity. Chapter 86. The First Genome to Leave Earth. In 2021, a mission launched quietly from Earth, carrying a synthetic DNA archive aboard a solar-powered probe. Its destination, Proxima Centauri, the nearest potentially habitable exoplanet. Inside the probe, encoded bacterial genomes for nitrogen fixation, algae genes for photosynthesis, human DNA, encrypted in triplicate, a CRISPR kit with instructions in 12 languages. This wasn't colonization, it was a messenger molecular time capsule, a whisper across light years that said, "We were here, we learned how to build life, we send its blueprints forward." It was the first spark of Crick and Watson's helix beyond Earth, terraforming with genes. By the 2040s, Mars was no longer a desert of dust alone. Genetic engineering made it a canvas. Scientists introduced cyanobacteria engineered to survive extreme UV and thin atmosphere, moss hybrids capable of growing in regolith and producing oxygen, heat-producing microbes to slowly warm enclosed biomes. The planet didn't change overnight, but the biology of Earth was now reaching into other world, smodifying, adapting, sowing new beginnings. Terraforming had always sounded like science fiction. Now, it was gene-by-gene architecture. Watson had once imagined genetic engineering for health. Now, it was being used to remake planets. The Interstellar Genome Program. Humanity prepared to travel farther beyond Mars, beyond moonstone exoplanets, reachable only via generational ships or cryosleep vaults. But bringing humans physically was risky. Instead, the Interstellar Genome Program, IGP, was born, launching missions with genomic blueprints, not bodies, sending embryos in stasis, or transmitting digital genomes to be printed on arrival by synthetic wombs. AI pilots would prepare environments. Upon arrival, human life would begin it as immigrants, but as new genesis. It was a bold reversal. Rather than humans traveling to stars, the stars would grow humanity. What is Earth if not a womb? Philosophers began reframing the idea of Earth not as the home of humanity, but as its birthplace, a fragile blue womb that nurtured consciousness, identity, empathy, genetic knowledge. And now, with DNA sequencers, gene printers, and adaptive biology, Earth had given humanity the tools to seed life elsewhere. Some feared this expansion. Others saw it as life fulfilling its biological imperative to reproduce not just within species, but across space. Crick and Watson gave us the alphabet of life. Now we were writing it on new pages in the cosmos. The helix in the galaxy. In a moon-based library lit by artificial dawn, a child opened a book. Its cover was dark and simple, spiraling lines embossed in gold. The child read about Francis Crick and James Watson, about how two men in a different century discovered something small and secret and how it changed everything. They read about base pairs, about CRISPR, about Earth's biosphere, about Mars gardens. And in the corner of the page was a quote, "In every helix there is hope, in every strand a story." The child turned the page, not knowing that their own DNA carried the names of these two pioneers encoded in a harmless stretch of junk DNA preserved for all who would come after. Part 19. Legacy Eternal. How Crick and Watson Rewrote the Destiny of Life. The line between genius and vision. In the shadow of history, Francis Crick and James Watson began as two brilliant minds with a shared obsession. What is the structure of DNA? They weren't the first to ask. They weren't the only ones to search, but they were the ones who saw the answer and dared to announce it to the world. They didn't have the best data, they didn't have the most refined tools, but what they had was relentless imagination, instinct, and an unshakable belief that structure determined function. From their model sprang the entire age of genomics. From their scribbles came therapies, justice, ancestry, and hope. They weren't just scientists. They were architects of possibility. From model to movement. The wire and cardboard model of the double helix built in Cambridge was crude by today's standards, but it remains enshrined in scientific history as one of the most consequential artifacts ever assembled. It wasn't just a molecule, it was a movement toward personalized medicine, toward a world without inherited disease, toward global ancestry awareness, toward engineered ecosystems, toward synthetic life, artificial minds, and biological expansion into the stars. Each step humanity took in mastering the genome was a chapter added to their legacy, and still, that legacy grows. The names inside the helix. In labs across the Earth and Moon, researchers have quietly encoded Crick and Watson's names into DNA watermarks in synthetic cells, quantum records of genomic simulations, protein folding AI's core parameters. They are no longer just names in textbooks. They are permanent entries in the code of life. Students extracting DNA from fruit in a middle school lab unknowingly hold their legacy. Patients receiving CRISPR treatment carry their story. Farmers planting drought-resistant rice use strands shaped by their discovery. The double helix is everywhere. So too are its discoverers. The helix, as a human heritage, at the United Nations Genome Museum, a glowing wall traces the structure of DNA 6 floors of illuminated base pairs stretching skyward in a glass atrium. Visitors enter and look up. At the top, inscribed in light, is the simple dedication to Francis Harry Compton, Crick and James Dewey Watson, who showed us the ladder of life and gave us the courage to climb. Asterisk, asterisk. People from all nations, cultures, and ages gather beneath this symbol. They may not know every detail of base pair bonding or helical turns, but they know. This is where the story changed. This is where science became destiny. This is where humanity began to know itself. A legacy still unfolding. Crick died in 2004. Watson, in his later years, faded from public life. But their work has never faded. It has echoed through every genome sequenced, every gene edited, every life saved. The double helix they revealed has become a spiral of knowledge, healing, controversy, power, potential, and its turnings continue endlessly, because life itself is a code, and codes do not end. They replicate, they evolve, they carry the message forward. As long as there is life, there will be the memory of the men who unveiled its alphabet. Part 20, final, The DNA of Tomorrow, a letter to the future, a message encoded. In a quiet lab beneath the Swiss Alps, a vial was sealed. Inside, a custom DNA strand, 300 base pairs long, sequence seemingly random, but decoded, it formed a message, not in English, not in any known human language. It was written in the universal code of life A, T, C, and G. It began, "To those who come after us, this is the language we found, written in every living thing. With it, we learned what we are. With it, we dared to change. We hope you use it, not to conquer life, but to understand it, not to perfect nature, but to protect it, not to become gods, but to become kind." Asterisk, asterisk. The code as compass. As humanity ventured outward into space, into AI, into synthetic biology, it brought the double helix as a compass. Not just a tool, a reminder that life is not made of grand statements, but tiny letters arranged with purpose. That evolution is not chaos, it is code unfolding. That healing is not magis, it is understanding. That identity is not fixed, it is written, rewritten, and shared. The DNA molecule became our collective mirror, and in it we saw not just who we were, but who we could become. Crick and Watson's enduring lesson. In a lecture hall in 2103, a teacher addressed her student shalograms flickering gently above their desks. She asked, "Why do we still study Watson and Crick? We've mapped the genome, we've rewritten it, We've seeded it in other worlds. A child raised his hand. Because they showed us the first sentence. The room fell silent. The teacher nodded. Yes, the sentence was already there, but they taught us how to read it. The spiral without end. DNA is more than molecule, it is metaphor. A spiral, a ladder, a thread connecting generations, a double helix winding through time. Watson and Crick didn't invent it. They revealed it. And in doing so, they gave humanity the most profound gift of all, a language for life, not just to speak to ourselves, but to speak to our future. Epilogue, a letter from the spiral. Dear future, you don't know us, but you carry us in every cell. We were curious, we were flawed, we searched, we found something small, spinning, silent, and in it we found everything. Use it well, edit wisely. Remember us not for who we were, but for what we tried to understand. The spiral is yours now. Climb, Francis Crick and James Watson, 1953 SIE.